Hello everyone, my name is Qinyang Shou. I'm from University of Southern California. Today I'm going to talk about my work of age-dependent changes of water exchange rate in blood-brain barrier assessed by diffusion-prepared arterial spin labeling. I have nothing to disclose. The blood-brain barrier plays a critical role in regulating delivery of oxygen and nutrients to the brain, clearance of toxic metabolics, and protection of central nervous system from infection. Recent studies found that the permeability of BBB is changing with age. As the age increases, the water channel on the perivascular spaces become disrupted, and thus the permeability of BBB will increase, leading to some dysfunction in the waste clearance in the brain, and will cause some diseases such as Alzheimer's disease. Among the ways to study BBB permeability, arterial spin labeling has the advantage of non-invasiveness and good reproducibility. So here we use the previously proposed method by Dr. Shou using diffusion-prepared ASL to measure the water exchange rate in BBB. The basic idea of this method is to use diffusion-prepared and non-diffusion-prepared readout to separate ASL signal from tissue and signal from capillary. The goal of this study is to investigate the relationship between water exchange rate, Kw of blood-brain barrier, and age. And also we want to compare the trend of the fitted CBF and ATT values by the proposed method with previous studies. Here is our imaging protocol. It used the same parameters as Dr. Shou used in his previous paper. We used this protocol to scan on several subjects from 4 different age groups, including pediatric subjects aging from 8 to 17, young adults aging from 23 to 29, elderly Latinos aging from 64 to 70, and African Americans aging from 40 to 81. All of the data were processed using a customized MATLAB toolbox with TGV regularization reconstruction of the raw data, and the SPA model was used to fit CBF, ATT, and KW maps. Regional analysis was performed by normalizing the brains to the standard AAL template. Here are 5 representative subjects from the whole data set of different ages. We can see a clear decrease in CBF, increase in ATT as the age increases. For the water exchange rate Kw, there is a slight decrease. This can be shown more clearly from the regression results from all our subjects. In CBF and ATT, there are significant decrease and increase with age, and in Kw, there is also a significant decrease with age. For regional analysis, we found in most cortical areas, such as frontal and temporal cortex, as well as subcortical areas, such as putamen and caudate, the fitted curve follows the global trend, which is increasing or decreasing linearly with age, while in some regions, such as parietal cortex and parahippocampal areas, there are some nonlinear relationship between those values and age, shown as the U-shaped curve of fitting. So our study found the trend of CBF and ATT values measured by diffusion-prepared Picasso is consistent with previous studies. Water exchange rate was found decreasing with age, suggesting compromising BBB function with aging. And the U-shaped curve may indicate the complexity of difference between brain regions. In conclusion, this is the first study investigating age-related effects of CBF, ATT, and KW using DP Picasso in a wide range of age groups. The results showed an overall trend of decreasing CBF, increasing ATT, and decreasing KW with age, although there were some variations across different brain regions. Thanks for your attention.